In this presentation, I'll briefly describe excitable tissue in regards to gamma-aminobutyric acid, benzodiazepine, and chloride in skeletal muscle. So what is GABA? GABA is a shortened name for gamma-aminobutyric acid, an amino acid, and its function is to prevent, reduce, or counteract neurotransmitters that work to produce an excitatory effect on the CNS. And this is synthesized in the presynaptic cytoplasm by an enzyme called glutamate decarboxylase. And this is transported via inhibitory transport vesicles where calcium binds to synaptobrevin, which allows these GABA-filled vesicles to fuse to plasma membranes, ready to be utilized. GABA binds through ligand-gated channels that cause an influx of chloride ions. Chloride ions diffuse into the presynaptic cells and are released by GABA receptors. GABA transmission contains a few receptor types, GABA alpha and beta, or GABA A and GABA B, and gamma. Chloride binds to GABA A. As the chloride ions come into the ligand-gated channels, the cell hyperpolarizes due to the negative charge in chloride affecting RMP, and thus causes inhibition of excitatory action potentials. In GABA B receptors, postsynaptic potassium is present and reduces the calcium conduct, which is also a way to prevent action potentials in the CNS. GABA can be found to involve many systems. For example, GABA can be found to decrease respiratory rate through its relaxing effect. Although GABA causes inhibition of excitatory action potentials, they do not, however, alter the firing of action potentials but inhibit them from reaching threshold through the hyperpolarization of the cell. BZD is short for benzodiazepine. It is inhibiting in nature and interacts with GABA A receptors. This binding causes changes in the chloride receptor of GABA and causes an inhibition of EPSPs. Benzodiazepine are allosteric modulators and work by altering receptor sites to produce a greater affinity to GABA. Benzodiazepines are used in the pharmaceutical company as a relaxer for the nervous system, and it's often used to treat anxiety. Just like benzodiazepine, alcohol has the same effect, and seeing as they both have the same effect, combination of the two together at the same time can cause serious reactions on the brain. Changes in behavior, mood swings, depression, and more seriously, impaired memory. People can often become addicted to the effects of these drugs with alcohol, trying to reach a high. So chloride also plays a role in skeletal muscle. Chloride channels are voltage gated and act on repolarization of the cell membrane by increasing resting conductance and reducing excitability. Much of this conductance takes place in T tubules, which contain chloride channels. The efflux of potassium causes the depolarization of these tubules. When mutations of chloride channels arise, channelopathies can occur, such as myotonia congenita, regularly spread along the cytoplasm. Myotonia occurs when chloride is inhibited and causes muscle stiffness, for example, not being able to release grip of an object. This is due to the protein kinase C, intracellular acidosis, and lactate ions, which inhibit chloride during muscle contraction. Here is an example of myotonia congenita in a goat, which is unable to relax its legs. Neurotransmitters that are inhibitory in nature act on the electrical charge of cells, causing an effect on the cell electrical threshold, causing inhibition or making a cell less likely to reach threshold for an action potential to create an excitatory effect.